Okay, guys, as promised, I am recording going through some exam questions for you to help you with your revision. Now, none of these questions are going to appear in your assessment tomorrow, but exam technique is quite useful to have at any time. So you can use this video anytime you want to help with your revision. Now, I have put the questions in your teams. Okay, so what I would do is I would pause this video if you haven't had a look at the questions, and I would attempt the questions first, and then I would go through and mark it using this video. Okay, now the first question I've chosen is nice and simple. Okay, it's a lovely diagram of a cell. Okay, and it's a multiple choice question. Now, with multiple choice questions, just be very, very careful because they can throw you and you might think it's really simple. Okay, so always read it properly. And it says, which type of cell does the diagram show? It says an animal, bacterium, a fungus, and a plant. Now, with multiple choice questions, you can always eliminate two. There's always a really silly answer. There's always one that might throw you a little bit, but there's already one that I can eliminate straight away, and that is bacterium, because I know that bacteria as a prokaryote don't have a nucleus, and all I can see straight away here is a nucleus, so I know that it's not a bacterium. Okay, I can eliminate that one. Okay, so I'm going to put a cross through that here for the purpose of this. Don't do this in your exam. Okay, so it's either going to be an animal cell, a fungus, or a plant cell. Okay. Now I can make an educated guess, okay, that it's not going to be an animal cell because it also has a cell wall. So I can eliminate that way. Okay. So it's either a fungus or a plant. Now I also know that I've got a big old vacuole in the middle. And I know that plant cells have vacuoles. So the answer here is going to be, okay, a plant. Always put a cross through like this, okay, in the little X in the box. Don't circle it or highlight it because that won't be picked up in your actual exam because these questions are marked by a computer, not a real life person, okay? Now we've got a question about membrane transport that again is multiple choice. It says a statement below describes the conditions required for some molecules to move into this cell, a concentration gradient and the use of ATP. Now, which of these statements is correct for the process of osmosis? Okay. Now, remembering back to osmosis, osmosis is the movement of water. Okay. So these statements are a concentration gradient and use of ATP. Now, it's saying, is only statement 1 correct? Is only statement 2 correct? Or is it 1 and 2? Or is it neither? Okay. Now, knowing what I know about osmosis, I know that it's passive. Okay. Now, passive means it does not require energy. Okay. So I can automatically rule out statement 2 because ATP is what we use for energy. Okay, ATP is the currency of energy. Okay. Now, osmosis moves from an area of high concentration to a low concentration. That is a gradient. Therefore, statement 1 is correct. Therefore, the only correct answer is 1. Okay, or A, 1 only. Okay. Now, moving on to some questions you've already seen. Okay. Now, technically, I'm making an assumption that there are 3 organelles in this question here. So it says the student uses a light microscope to look at the human cheek cell. The student makes this drawing of the cell, name the organelle shown in the drawing. Now, technically, I have an organelle here and I have an organelle here, but the main one that I want to focus on is this one. Okay, this is my organelle that I am identifying, and we should all know that that is the nucleus. Okay. Then we are moving on to a magnification question. Now, here they have not given you the exam equation. Now, they might give you the equation. I've seen some questions and you've seen some questions where they've given you the equation. So it might be good to write down on your sheet or front of your paper just the equation triangle so that you can remember it. Okay, so remember we're using I am. Okay, so I is image, size, A is actual size, and M is magnification. Okay. Now it's telling you here, okay, that the mitochondrion too small to be seen using a light microscope. The drawing shows a mitochondrion that's been magnified, and it's giving you a size. Giving you the size of it is 60 millimeters. Okay. And it's saying here the actual length is 6 micrometers. Now calculate the magnification of this drawing. Now when we do our calculations, you will get 1 mark for converting the units. Now the units need to be the same. Okay, now I can automatically say that the image size that they've given of the picture is in millimeters and my actual size is in micrometers. So I've already got a disparity. Okay, so the first thing I want to do is convert my 60 millimeters into micrometers. Okay, so what I'm going to do is I'm going to do 60 times by 1,000 because there are 1,000 micrometers in 1 millimeter, and that gives me 60,000. Okay. The second thing I need to do is then put it into my equation. Now it's asking for the magnification. Now that means from my triangle, I need to do image size divided by actual size. So I just need to do 60,000 divided by 6. And that is 10,000. You get a mark for the conversion of the units and a mark for them putting it into the equation, substituting it into the equation. Okay, so they haven't given you the equation here. You have to remember it. Okay, so I would always get into good habits of remembering that equation and remembering how to convert down to your units. So remember, if I'm going from millimeters into micrometers, then into nanometers, every time I'm going into a smaller unit, I'm multiplying by 1,000. Okay, because I'm going to have a bigger number. If I'm going backwards, I divide by 1,000. Okay. So then we've got another maths question. Now this is only 1 mark, it's multiple choice, but you've got to do a bit of working out first. Okay. Now it says here, the table gives information about mitochondria in different human cells. Okay. And it says you've got the mean number of mitochondria. Oh, sorry. I clicked on highlighter there. The mean number of mitochondria per cell, the mean volume of the cell in micrometers cubed, and it needs to know the mean number of mitochondria per micrometers cubed. Okay, so what I need to do, okay, is I need to do 5,000 divided by 15,000. Okay, and that should give me the answer. Okay, so if I do 5,000 divided by 15,000, I get 0.33 recurring. So the only correct answer there is A. That's one of my answers here. Now, then you have, okay, a question that says, comment on the differences in the data for the sperm and the egg. Now, comment on means that I need to use the information that I've got here and then give possible reasons why that might be. So, for example, okay, using the data that I've got for the sperm and the egg, so I'm looking here now. Okay. From the volume, I can automatically tell that my egg is much larger than my sperm because the volume of my egg is 4 million, okay, micrometers cubed compared to 30. Okay, so that's already 1 mark I've got here. I only need to give 3 points here. Okay, so I can say that my egg is much larger than the sperm. Okay, I can also say that there are more So I was just doing a bit of data analysis there. More mitochondria per micrometers cubed in the sperm, because if you're thinking about it, which one of these cells has to move? This one does. Okay, my sperm has to move to the egg. The egg doesn't have to move to the sperm. So if you're looking at the ratio, okay, per volume, okay, there are more mitochondria in my sperm cell compared to the volume of the cell, okay, because the sperm has to move. And to move, it requires energy, therefore it needs mitochondria. To the egg. All right, and that's how we answer that question. And now we've got good old Chlamydomonas that you guys all hated when we did this in class, because you were all in uproar that I asked a question about something that you had never seen before. And this is what we call an application question. Now, an application question is where we teach you a specific concept and we ask you to then apply that concept. Okay. Now, you apply in an unfamiliar context. Now, being able to apply your knowledge to a familiar and an unfamiliar context, okay, is a grade 7, grade 8 skill. Okay, so that's what we're aiming for here. Now, the structures labelled A and B can automatically see that B, okay, looks like a little oval shaped or bean shaped thing with a squiggle in it. We automatically know that is a mitochondria. Okay, so B is mitochondria. Now, the rest of this is labelled. So I know that A, it's not going to be a vacuole because I've already got that labelled. A must therefore be the other big thing that's in a cell, and that's the nucleus. Okay. The next question says, Chlamydomonas has some structures that are normally found in plant cells but not found in animal cells. Name 2 of those structures. So here we're looking at organelles that are only in plant cells, and they're already labelled for me, so I can already say cell wall and chloroplast. You could also put starch grain, because starch grains are only found in plants, because we do not store our glucose as starch, we store it as glycogen as animals. Okay. Now it says, state why Chlamydomonas is not classified as a plant. Now that is where you need to know your features of your kingdoms. Okay, so for your 5 kingdoms, we are talking about plants, animals, fungi, prokaryotes, and protoctists. Okay, those are our 5 kingdoms. Okay. And we need to know the main features. Now, this is saying, why is it not a plant? Now, one of the main things is all plants, okay, are multicellular. And this, okay, is single-celled. It says it in the question, okay? All right, so always read the question twice, okay? So because it is not multicellular, okay, it is not a plant. Okay, tells you it's a protoctist, okay? Now, it does say, explain how Chlamydomonas obtains energy. So where does it get its energy from? As a similar to a plant. It's similar to a plant because I know that it's got a cell wall, it's got a vacuole, it's got a chloroplast. Okay. All right. So Chlamydomonas obtains its energy by using its chloroplasts. Okay. So chloroplasts, what is the job of a chloroplast? So where you're looking at your function of organelles here. Okay, chloroplasts absorb light. That's one mark. To be used in photosynthesis. Now, it also then says Chlamydomonas has a flagella, suggests a function of these flagella. Okay, so when we briefly looked at prokaryotes, we looked at flagella, and also sperm have a flagella. What do sperm use their flagella or their tail for? That is movement. Okay. Okay, swimming would also be accepted as well. Okay. So now we're moving on to surface area to volume ratio. Now I'm doing this one, you haven't got a calculate surface area to volume ratio exam question on your assessment, but I thought it would be good practice because these do come up a lot of the time on your final exams. So we do need more practice with this. Now, as we go through the course, we will have synoptic assessments, so there might be questions from previous topics on your assessments as we go through. All right. Now, this question here, I've got 3 cubes. I've got cube A, I've got cube B, and I've got cube C. Now, it says here that the student used a method to investigate the movement of molecules by diffusion. Now, I did this as a little video. So they cut 3 different size cubes from a block of clear agar jelly, and he tells me that cube A has 2 centimeters by 2 centimeters by 2 centimeters. Cube B has sides 1 by 1 by 1. So I've got 2, 2, 2. Cube B, I've got 1, 1, 1. And here I've got 0.5, 0.5, 0.5. Okay. So he placed each cube in a beaker of a red dye for 5 minutes, removed the cube from the dye solution, washed the surface of each cube with water and dried with filter paper, and then he cut each cube in half to examine the newly cut surfaces. Okay. Now, the first question has nothing to do with this little bit of the blurb at the top, but the rest does. Okay. So the first one does say state what is meant by the term diffusion. Now, this is only a 1 marker and it's state, which means you only need to give it. But if I need to know what is meant by the term diffusion, I need the definition. So you need to give me the actual definition for diffusion, which is movement of particles, you can say substances, okay, or molecules or ions, whatever you want to say, movement of particles from a high concentration to a low concentration. You can even say down a concentration gradient, sorry, not a high concentration gradient, down a concentration gradient, or you can also say from an area of high concentration to an area of low concentration. I'm being lazy and I didn't want to write any more words. Okay. Now, that is the definition of diffusion. We're investigating diffusion. Okay. So the next question is linked into the investigation at the top. It says give 2 factors that the student should control in their investigation. So the 2 factors they should control will be anything that could affect the rate of diffusion, because that's what we are going to be investigating. Therefore, factors that affect the rate of diffusion, I might need to think about. So factors that might affect it could be temperature. Temperature affects the rate of diffusion. If I increase my temperature, I increase my rate of diffusion because my particles have more energy. Okay. It could be concentration of the dye. I've got a greater concentration gradient outside than I do inside, which I do. Then therefore my particles will move faster. Okay. The thickness of the agar will affect it, because if I've got a thicker agar to move through, that will slow down my rate of diffusion. Okay. So diffusion distance is one. So I need it to be really thin to increase my rate of diffusion. Okay. Diffusion distance, and the last one is the volume of the dye. If I've got more dye, then more can diffuse. Okay. Now, all of those things are going to affect the rate of diffusion, but it's not what we are actually investigating. Here we're investigating the surface area to volume ratio and how that affects the rate of diffusion, okay? But the other things that could affect it are things we need to control. So then it asks you to do a bit of maths. So it asks you to calculate the surface area of cube A. Right, now cube A is 2 by 2 by 2, okay? But to create the area or calculate the area, I need to do, okay, base times height. Okay, so I do 2 by 2. That gives me 4. Did have to think about that for a second. Okay, it is 5:30. Now, how many faces does a cube have? Okay, because we're looking at surface area, total surface area. Okay, so I want to do the number of faces times by the area of one face, okay? And cubes have 6 faces, therefore I do 4 by 6, okay? And I do 4 times 6, and that gives me 24, okay? So I've got 24 centimetres squared, okay? Now, then I've got a question here, don't forget this one. It's hidden underneath this, okay, because you haven't got much space, but it says complete the table by calculating the surface area to volume ratio for cube A. They've already done the volume, but if I'm calculating the surface area to volume ratio, I need to get it as something expressed to 1. So I want to have x to 1, okay, and 1 is going to be my volume. Always do it, surface area to volume, okay, so my SA to V, okay, I want to have something to 1. Therefore, if I divide my surface area by my volume, I am going to get that. So if I do 24, which is my surface area I've calculated, and then my volume is 8, if I do 24 divided by 8, I get 3 to 1, and I can put that in here. Okay. So the diagram shows the newly cut surfaces of the cubes. Using a ruler, determine the distance the red dye has diffused into the cubes. And what you need to do here is just measure. Okay, you just need to measure it. Okay, so what the mark scheme is telling me here is 2 millimeters. This will differ depending on the size of your print for your question, so always do it. Okay, so they will accept a range of 1 to 2 millimeters. Okay, I'm just going to put 2 just for, you know. Yeah, it is 2. Okay, for each of them. But they do offer a range for human error, okay, because it's only 1 mark, okay. Now, here comes the tough stuff. So this, okay, is where we are looking at applying it. So it says use the data from the table and your measurement from the part before to comment on the effect of size on transport in an organism. Okay, now it's 4 marks. Okay, so we're commenting on the effect of size. So commenting, so we're just going to look at the data first. So we can get some marks just by looking at the data. So if I look back here, okay, I can see that as my cube gets smaller, my surface area to volume ratio gets larger. Okay. Now, from the data though, all of my cubes, so here, all of the cubes, the distance is the same. Okay, so I can get a mark just by saying that. Okay, because when I measured all of them, they're all the same distance. So the dye has moved into each cube the same distance. That's 1 mark, that's 1 bullet point. That's one thing I can get from the data. Okay. However, when I look at them, okay, there's a lot of empty space that the dye hasn't filled in cube A. Okay, because it's got a much smaller surface area to volume ratio. In cube C, I've only got a tiny space left in the middle here that the dye hasn't penetrated. And I can use that in my answer as well. Okay, so I can say, but cube C has a greater proportion of penetration by the dye. I know that's not a nice word to use, but that is what has happened. Okay. Okay, the dye has moved in further because it's a smaller cube. Okay. Now I can therefore see, okay, from my table that as I get smaller, as my cube gets smaller, okay, my surface area to volume ratio has increased. I also get a mark for talking about that, and that's all information from the table. So far, I haven't used any of my own knowledge. I've only used evidence from the table or the information that I've got that I can see. So I've used the pictures, I've used my measurements, and I've used the information in the table. Okay. As size decreases, surface area to volume ratio increases. Okay. Now my last point is interpreting that. Okay. So I know that if I've got more of my dye that's moved into my smaller cube, okay, say for example my dye was not dye, it was oxygen. I've got more oxygen that's moved into that space in cube C compared to cube A. Therefore, my smaller organisms, okay, are much more effective at using diffusion alone as a transport substance. So, for example, transport substance, transport method, that's the word I'm looking for. Okay, so that is a point that I can use to explain. So smaller organisms are more efficient at carrying out diffusion. Okay, so the next question, the last question, actually, yeah, no, it was the last question. The last question is talking about osmosis. I couldn't find any questions on enzymes. I did want to find some for you, but I couldn't find any questions on enzymes, okay, that weren't ones that I would need to use later on or have already used or are on your test, okay? So this question, again, very, very similar to the one that we had, but a little bit simpler, okay? I've got here apparatus that shows osmosis, okay? Now it says here that I've got a glass tube, sucrose solution, which is inside. So my sucrose solution is inside my little, it's called a thistle tube, okay? I've got water surrounding it and I've got a partially permeable membrane covering it, okay? So what I'm going to be comparing here is what is going to happen inside here comparing to outside, okay? Right. Now it says explain what happens to the level of sucrose solution in a glass tube. So you need to tell me what's going to happen, first of all, and then you need to tell me why. Now, you only get 1 mark for telling me what's going to happen, but then the other 2 marks come from why. Okay. Now the first thing that I'm going to do is I am going to say that my water, okay, is going to be 100% water. There's no sucrose in it whatsoever. My sucrose solution, okay, it hasn't given me a concentration, okay, but I'm going to say that it is less than 100%. Okay, that's the only thing that I can make out because obviously I'm going to have sucrose in there. There's less water in there. Okay, now using that information, I can assume that water is going to move into my sucrose solution because that is more concentrated. Okay. Now, if water is going to move in, what's going to happen to the level in my tube? Okay. If water moves into something, what's going to happen to the volume of water in there? It's going to increase. Okay. So my water or level of sucrose, sorry, I'm not going to say water, I'm going to say level of sucrose because that's what my question has asked me. That's what it says here. Okay, so my level of sucrose will move up the tube. You can say increase. Write that in brackets. And now I'm going to say the because bit. Okay, this is because water is going to move in here. Water moves into the tube. Okay. Actually, I'm going to write that a bit better. Water moves across the partially permeable membrane. Okay. Because the sucrose has a lower concentration of water. That is your 3 marks. Okay, you can explain that the water has a higher concentration and therefore your water will move from there into there, but I think you've answered that in those 3 marks there. If it was 4 marks, I would definitely go, this is due to osmosis. Always mention osmosis if you can. Okay, now B then says, describe how this apparatus could be modified to measure the rate of osmosis at different temperatures. Now, essentially, what I need you to do is, what can I add to this practical setup to investigate or measure the rate of osmosis at different temperatures? Okay, so if I need to measure the rate of something, I need to measure the speed. Okay, so speed. Okay. So if I need to measure the speed, say for example, I needed to measure the speed that I would run at, thinking about what I would need to measure the speed, I would need to know how far I'd travel, so I need to know a distance, and I would need to know how quickly I covered that distance, so I would need to know time. Okay, so thinking about distance here, how can I measure distance here? The easiest thing is to add a ruler, add a scale to my tube. Okay, so I would need to have a ruler, okay, to measure distance. So you need to tell me what I'm using it for. A ruler to measure the distance. I would need something to measure the time, so a stopwatch or a timer. Measure time. These are all additional pieces of equipment, or how this equipment can be modified, how I can add to it to calculate the rate, or measure the rate, sorry. And I would need to then, to change the temperature, I would need something that was able to change temperatures. Okay, so how can I easily change temperature? I would need to add heat. How can I add heat to a piece of equipment? I could use a Bunsen burner. Add heat. Or I can add a water bath. Okay, so I'm going to add that, or a water bath. Either one would be acceptable ways, okay? Hasn't necessarily said that you need to keep it at a constant temperature. If it did say to keep it at a constant temperature, so for example, it was saying how could I modify or improve this method to keep it at a constant temperature, then I would say a water bath, okay? But that is how you answer that question, okay? Hopefully that has been helpful for you, okay? And if you have any questions, just drop me a message on Teams or an email, or come and see me tomorrow before your test.